Welcome to this next part of the NeuroExpress tutorial. Here I would like to demonstrate the spike train analysis module of the program. For those who are not yet familiar with NeuroExpress, let me just give a brief introduction. This is basically a Windows program designed for the analysis of various electrophysiological data, including voltage step responses of neurons recorded in whole cell configuration, spontaneous postsynaptic currents, and trains of action potentials. The spike train model that I introduce here will work with membrane potential data or spike arrival time data. The program can analyze single and multi-channel recordings such as those recorded with planar multi-electrode arrays or brain implanted electrodes. The recording here shows the firing pattern of a neuron in a primary cell culture. The program detects the action potentials and calculates the firing rate, interspike intervals, ISI histograms, Fourier spectra, Poincare maps, and many more metrics and graphs, and does it pretty much automatically. This is a single channel recording containing a few hundred spikes, but the program can easily handle millions of spikes in multi-channel recordings. The next example shows the firing output of 4 molluscar neurons recorded simultaneously using sharp electrodes. The voltage traces are not shown here, only the spike arrival times are used for the analysis. The middle graph panel shows the entire recording, where you can select ranges of interest to analyze. You can use the mouse to define a window of interest, and then you can drag this window and the corresponding graphs will be updated instantly. Next is a spike train recording of 6 neurons in the stomatogastric ganglion of the lobster. These are robust bursting neurons in the gastric network, so the spikes appear as discrete bursts repeating in a semi-regular manner. The program detects the bursts automatically and calculates the burst cycle period, intraburst spike number, duty cycle, and several other parameters. The spike rate here is represented as a color map of spike density function for all 6 neurons. The spike density is a smooth and continuous estimator of the firing frequency based on the convolution of the spike times with a Kernel function, such as a Gaussian or Henning window. In this example, we can easily see the fine structure of the gastric oscillations and the presence of the faster input caused by the interaction with the pyloric network oscillation. This is especially obvious for the middle neuron, which is called the lateral pyloric gastric neuron. Okay. Well, the Fourier analysis of the spike density is very useful because these graphs will show the periodicity and regularity of the network oscillations in this network. But they will also show the frequency modulation arriving from the faster pyloric neurons. These are shown by these peaks around 2 Hz in the Fourier spectra. These are not FFT graphs. The program uses the traditional slow method to calculate the spectra, but the speed is still impressive. The spectra are updated almost instantly as you can drag the zoom window. I also demonstrate here a method to characterize the correlations among these neurons. The program performs pairwise comparison of the spike density functions and calculates the correlation coefficients. This is also very fast. You can see that these graphs are updated immediately, and neurons that tend to fire synchronously will be represented by the red colors. Colors toward that, so they indicate the more positive correlation coefficients. Blues are the opposite. Okay, now we arrive to the analysis of multi-channel recordings. These spike time data were collected from 48 well MEA plate where each well contained 16 electrodes to record neuronal action potentials. Obviously, tens or hundreds of thousands of spikes can be recorded in cultures displaying strong network activity. This is a fine example of that. The upper right panel represents the map of the 48 wells, and the colors indicate the overall level of spiking activity. You can easily switch between the wells just by clicking on the corresponding circles. So you can see that there are wells with lower activity, lower number of action potentials, and the red one indicates the strongest or highest level of network activity. The spike density functions for all 16 electrodes are calculated, and you can clearly see the degree of synchronization across the recording channels. You can also find brief bursts of action potentials and more prolonged episodes of elevated activity called superbursts, like here. So these are very intense and prolonged episodes of firing in the network. Smaller bursts are also easy to find, like here. These are beautiful examples of bursting in primary cell cultures. The correlation matrix here is also calculated for the data. Now we have a total of 240 pairwise comparisons. Again, these graphs are instantly updated as you select the range of interest. You can see that the strongest correlation exists between these two channels, indicated by the yellow ellipses, and this correlation and the strongest correlation persists as we move across the entire recording. So this is an interesting example where the strongest correlation is between two channels in very close spatial relationship as well. Synchronization across neurons can be also analyzed by a matrix called burst synchrony index. Here we can see the temporal relationship between the bursts of the compared neurons and how tightly they fire together. The colors indicate the order of the burst onsets, and the size of the squares indicates the temporal precision of the burst coupling. And you can also go across the entire recording and analyze how conversed these burst synchrony indexes, the indices, are. Okay, so just a few more nice examples of this burst synchronization for different wells. The next example shows 1 hour of firing activity in the 48 well plate, and now we have more than 300,000 spikes recorded in well number 28. Still, the navigation across the recording is pretty easy, and the interspike interval plots and symbolic histograms are updated smoothly. NeuroExpress has now this new feature that allows the quick calculation of symbolic histograms that can be a powerful tool in spike trend analysis, especially when effects of drugs or drug candidates are tested in MEA plates. We can set many parameters of the calculations, such as the maximal intraburst spike time or interval, or the smoothing parameter for the spike density estimation. The results of the analysis will be affected by these parameters, so careful adjustment of those is certainly recommended. When all input parameters are set and all the calculations provide accurate results, then we can export the calculated output parameters into Excel. We do this by clicking on the Send button. We will obtain firing rate, burst cycle period and other parameters for each electrode in the well, and we can export the results for all wells that contain useful data. So next, like this, send again, send again. So this is a convenient output of the calculations from NeuroExpress, and then you can select the features and parameters that you are most interested in and perhaps ignore the others. The description of all these parameters can be found in the documentation, and also you can export a brief description of the parameters into the Excel. So the program can actually explain which parameters mean what. So if you are interested in trying out this program, then you can visit my ResearchGate page and download the trial version of this program. You can find this page by googling NeuroExpress download ResearchGate, and here in this list you will find some documentation and some general information on the program. But the link to the download is this one, Analysis of Miniature Excitatory Postsynaptic Currents, and then you will see download of the program. This is actually not a PDF file, but a ZIP, a compressed archive that contains the executable and a few sample files. You can unzip uncompress this file and copy it into a directory like under program files, and then you are ready to start and run the program. It will not require anything else like a runtime library or some additional component like a Python environment or MATLAB. It's a standalone executable, and you can use it immediately after downloading.